So next up, we have Dr. Kurtzberg, who is an internationally renowned expert in pediatric hematology, oncology, pediatric blood and bone marrow transplantation, umbilical cord blood banking and transplantation, and novel applications of cord blood and birthing tissues in the emerging fields of cellular therapies and regenerative medicine. Dr. Kurtzberg serves as the director of the Marcus Center for Cellular Cures, director of the Pediatric Transplant and Cellular Therapy Program, director of the Carolinas Cord Blood Bank, and co-director of the Stem Cell Transplant Laboratory at Duke University in Durham, North Carolina. Today, she'll be providing us an update on the activities of the Krabbe Newborn Screening Council, which is sponsored by Hunter's Hope. Thank you, Dr. Kurtzberg.
Thanks, Margie. I'm going to share my screen and hopefully this is going to work. And I need you to tell me if you see, hold on one sec, one slide or two.
We see two.
Okay. Now,
hopefully
we see one. Okay, yep. Great. Well, it's a pleasure to be here. I'm sorry I'm not with everyone there in person. I realize none of us are, but hopefully by next year, we'll be able to meet again in Buffalo. I'm going to give an update today on the Krabbe Newborn Screening Council, but also give a little bit of background, somewhat summarizing and consolidating what Dieter just went through. So I first want to acknowledge that really all of this work on newborn screening for Krabbe disease started in New York, and really they did an enormous amount of work that provided us with critical information that's moving the field forward. In their initial work, a risk assignment was designed for screen positive babies, but in retrospect, we learned that that was too broad and identified babies who we called at risk, but were actually not affected with or at risk for the development of Krabbe disease. The initial protocols that were designed for follow-up of these quote unquote at-risk babies was quite invasive, required very frequent follow-up, and as such compliance was poor. And there was no way for third-party payers to actually cover the costs of the follow-up. And there was valid criticism expressed regarding these initial efforts, but I'll say that without them, we wouldn't be where we are today. And as you've heard from Dieter, the recognition that cycosine could be measured on newborn blood spots and that it correlated with activity of disease, particularly in infantile Krabbe disease, really was a revolution and changed the field. Babies with infantile Krabbe disease, or as it used to be called, early infantile Krabbe disease, have a positive screen and then also have a cycosine of more than 10 on newborn blood spots, and they can be rapidly identified and referred after parental counseling and agreement for transplant or in the future gene therapy. And I'll just say that from what we know today, early urgent referral for these babies is critical, whether or not they're being treated with transplant, gene therapy, or both. Babies with later onset Krabbe disease will have a positive screen, and in many cases, an intermediate cycosine between 2 and 10. And those babies can be identified and followed based on a risk stratification that Rob Stone created with a team from the Newborn Screening Council. And unaffected babies who will typically have a cycosine under 2 won't require additional follow-up, although there are rare babies identified where cycosine is low, screen is low, positive. And when mutation analysis is performed, there's a combination of pathogenic mutations, or one pathogenic mutation with a variant of unknown significance, additional follow-up may be required. Most newborn screening labs, as you've heard, have incorporated cycosine into a second tier or reflex testing, but the actual way that that happens differs from lab to lab, and the timing of that also differs from lab to lab. You've seen Dieter show how important cytosine is in identifying babies with infantile Krabbe disease, shown in the upper left. And this is cytosine in the newborn blood spots, or at risk for later onset Krabbe disease in the second bar. But I want to point out that as the field's evolved, and as he mentioned, cytosine can also be mentioned in measured in red blood cells. And the value, the absolute value of cytosine in red blood cells and the absolute value of cytosine in newborn blood spots are not the same. So we all have to learn more about what these numbers mean, which number is being reported, and how they should be followed. So you've heard, but it's important to mention again that babies with infantile Krabbe disease require immediate counseling of the family and referral to a transplant center. And these babies have a positive screen and a cytosine over 10. And this is on the newborn blood spot. Babies at risk for later onset Krabbe disease and require long-term follow-up and monitoring, and they will have a positive screen and a cytosine on the newborn blood spot of 2 to 10. And in those babies, mutation testing should be done, and there is time to do it to further determine their risk. And then babies can also be unaffected either with polymorphisms or other genetic abnormalities that make their enzyme level low, but where they really don't have Krabbe disease and no follow-up is required. Rob identified or defined in his paper that for the babies at risk for later onset Krabbe disease, we can divide them into a high-risk group and a low-risk group, and follow-up pathways, which are inherently more aggressive in the high-risk group than the low-risk group, are defined. As an update for newborn screening, as you've heard, there are 10 states performing newborn screening for Krabbe disease today, and more than 6 million babies have been screened. There also are 3 companies developing gene therapy for Krabbe disease, 2 of which are in clinical trials right now. In one case, the gene therapy is administered after transplant in babies with infantile Krabbe disease, and in the other, it's administered as a single therapy or standalone therapy without transplant in infants with symptomatic Krabbe disease. And most importantly, the RUSP application was resubmitted and is currently under evidence review. So because of all these advances, we created a newborn screening council for Krabbe disease, and we created it for multiple reasons, but which include to assess policies and recommendations around newborn screening and follow-up, and to serve as a resource for the newborn screening labs and doctors who take care of these referred babies to review complex cases and to determine the best path forward for these babies. The goals are to have all newborn screening labs enable cytosine testing with rapid turnaround, meaning 2 days or less, although we aren't quite there yet. We also hope that each state will develop a predetermined roadmap for referral of infantile Krabbe patients to transplant centers for transplant, or in the future gene therapy centers. To develop guidance for identification of babies at risk for later onset Krabbe disease, and stratification and follow-up protocols, which Rob's paper already defines, but we know we'll have some modification as we learn more in the future. And then finally, we hope to develop care maps for rapid referrals for transplant or gene therapy for babies with the infantile form of the disease. Joe Orsini and I co-lead the Newborn Screening Council, and Grantham is the administrative director. And the members include members from newborn screening labs, transplant centers, laboratory experts, clinical experts, neurologists, geneticists, et cetera, and clinical gene therapy centers. We've had about 21 meetings to date, mostly virtual, and we've reviewed more than 10 unusual cases. We do have industry participants, but because of concerns of confidentiality about patient information, we now only have certain meetings where the industry participants are invited, and they occur on a sporadic basis. So the purpose is to provide continuous improvement of newborn screening for Krabbe disease, both for methods and follow-up algorithms, to update and improve newborn screening results and how they're communicated, to help orient new states participating in newborn screening for Krabbe disease to learn from our past and what we know for the future, and to optimize laboratory methodology for screening. We are continuing to evaluate changes to recommended follow-up protocols. We are a forum to discuss difficult to diagnose cases. And as you've heard from Dieter, there are many of those as more and more mutations are identified. And we really have to learn what they mean, not for the family as well as for the community. We hope to be a platform to discuss any and all issues related to screening for Krabbe disease and to provide feedback on proposed changes to screening. And to ensure all members are informed of the latest improvements. And then to continually keep an eye on the status of gene therapy or other novel treatment intervention protocols. To date, we've had projects that include 2 manuscripts that are both published. One is early results of newborn transplants in the modern era of screening, and the other is diagnosis of late onset Krabbe disease and guidelines for clinical follow-up. We've been involved with many others in putting in the RUSP application. I think one of the most important things we're doing now is continuing to provide consultative support for difficult cases and continuing to educate providers who transmit the newborn screening results. And I'll be talking about that in about 15 minutes again. We hope to improve methods of communication of newborn screening results and support for families whose babies have been diagnosed with infantile Krabbe disease or at risk for later onset Krabbe disease. These are the current members of the task force or the advisory council. And again, as states add Krabbe to their screening panels, they are invited to join the newborn screening council. I'll stop